So it's a real pleasure for me today to introduce Susanne Rafelski. She is the Director of Assay Development at the Allen Institute for Cell Science. And yeah, I feel really embarrassed. I have to go back to the paper here. We work already 4 years. It was always so pleasant and high quality conversation, so I never asked her for her credentials. So before joining the Institute in 2016, Susanne was assistant professor in the Department of Developmental and Cell Biology, the Department of Biomedical Engineering, and the Center for Complex Biological Systems at UC Irvine. So she is really interested in deciphering the complex patterns which governs the rules of cells and life. Yeah, Susan, I can guarantee you're in for a really fast-paced treat. Welcome, Susan.
Thank you. Good morning, everyone. I'm Susan, and I want to say what Winfried said, which is that we're really, really happy to have you all here. The Allen Institute, has a mission to do open science and to interface with the community, and we actually really love bringing the community to us so that we can do that and going out to the community. So we're really happy to see you. So at the Allen Institute for Cell Science, we want to understand the cell. We want to be able to look at the cell and know what it's doing, what it's going to do, and what it has done, what it did, and what it will do. I should follow my slides. And we think that we can do this because of the very tight interplay between cell organization and cell function. If we can watch all of the main structures in the cell, figure out their positions, their dynamics, their morphologies, we think that we will be able to develop a signature of what a particular cell is in its state, as well as its past and its future. So we have several really big basic science questions we're interested in at the Institute. We want to figure out how to define and categorize cells to create a meaningful and useful cell atlas. We're really interested in the principles of cell organization, and we're very interested in how cells transition between states. So we can expand these questions in the context beyond basic science, also of differentiation and regeneration, where especially these state transitions are incredibly important, and of course, how this then impacts research and disease. So our approach is an image-based approach. We think that we can learn a lot from what's inside the cell and watching it. We also take a holistic approach. We want to think of the cell as a whole and all of its parts, not very particular subsections of the cell. And so we do, our main work is 3D live cell imaging, mostly at high resolution, to get that sort of information. And a picture of all these parts in the cell at one moment in time, that's sort of our signature of that functional or transitionary state that the cell may be going through. And in that way, we want to create the state space of human stem cell signatures, but we also want to understand that space. So our model system is the human-induced pluripotent stem cell. And more specifically, its differentiation process into the differentiated cardiomyocyte, which is a really wonderful, well-established, and reproducible differentiation process. The human-induced pluripotent stem cell, of course, has all of this potential to be differentiated into many different cell types and to be of use both as a model system for human development and for regeneration purposes. So we really think that's a great place to be. We want to be able to study ourselves in as normal an environment as possible. So we want to watch the parts of the cell, and we want to do so without perturbing the cell. And we already committed to the stem cell, which is as normal a behaving cell as we can use. So we do endogenous gene editing to tag our structures using CRISPR-Cas9. And we spend a lot of time with a lot of quality control to generate these cell lines that we hope are as unperturbed as we can get them for our purposes and yours. We also spend quite some time developing differentiation protocols that are reproducible and scalable. And we then, of course, want to have ways to visualize our cells, which includes generating a reproducible pipeline for imaging in 3D for live cells, as well as assays that we need to develop, depending on our use case, to be able to integrate the cells and have a reference system for those cells. Which brings me to this integrating, this holistic approach, which is really a centerpiece of what we do. We fundamentally believe that it's important to think of the cell as a whole and to think of all of its main parts, to look at structure X and structure Y that usually people may not look at together in the same context. But we also want to integrate beyond structural organization. And we are using assays for gene expression, including single-cell RNA-seq and RNA-FISH, to start tying together these cellular organizational signatures with gene expression signatures. Everything we do at the Institute has a purpose to be, in the end, quantitative, analyzable, and hopefully modelable in some way. We generate models that range from biophysical models to statistical machine learning type of models that try to encompass all of the variation in our cells. And we are building platforms for image visualization, interrogation, and model output because we're so committed to wanting to have this be open access for all in a useful way, everything from our data to our tools to our interpretations and to yours. We are started with the undifferentiated cell. And we are also starting on this end with the differentiated cardiomyocyte, and we're filling in the space in between. So today I'm going to talk to you mostly about the undifferentiated HIPSC, but know that we are very excited about steps towards understanding this as a full process in the same way that we're doing for these end states. Not really end, they keep going, but. OK, so this is our institute. This picture is about a year old now. We've been making cells and imaging them for 3.5 years. We're about 65 people. And when you put 65 people together and say, OK, all of you work together on one project, that's beautiful and wonderful and generates a type of science that I certainly hadn't seen before, which is this big team science that allows us to try to do work and projects in a way that an individual investigator would not be able to do, but also in a way that will then empower those investigators to join in with us and do the science together, which is this open science and community engagement part. Again, if you take 65 people and put them all on one project, you can't just stick them all in a conference room and say, OK, go. It helps us a lot to have a little bit of organization. So we have several teams, and those teams do have some specialties. And we have this hypothetical, beautiful, linear sort of flow of information through those teams, but know that the reality is that we all interact and cross-interact with each other all the time. I want to put a special shout out to our software development team, which has to tie all of us together in a way that we can actually freely exchange our data, our findings, our tools, and all of our huge amount of information and metadata that we're trying to track to make sure that we do this in a careful way. So if I do one thing today, if I succeed with one thing, it's to try to get you to this website. This is our portal. And it's worth exploring. It's worth exploring in depth. There are many different options in here because you have access to everything that we can put out there at any moment in time. We work really, really hard on that. And so I really hope you go and visit and check it out. One of my favorite new pages on this portal, on allencell.org, is actually related to visualization driving science. So if you take a 3D image and you try to look at multiple channels and all of the Z slices at once, you get something that looks sort of like this. And when you start thinking about how to do better visualization, this is just visualization. This is not changing the data directly. Via Agave, which is a path trace rendering tool, then you can get images like this, where you get the spatial relationships between some of the structures in the image in a much higher clarity way. And it works online on all of our data, and you can download it and use it on your data, and it's spectacular. It took my breath away when I saw it. So I encourage you to take a look. OK, so I'm going to go through a couple of a little bit deeper for each of these steps and what we do. I'm going to start with the gene editing. So this is our workflow. We've had this for a while now, so a lot of people know it. When you do CRISPR-Cas9 and gene editing to stick GFP endogenously into the genome, we generally get monoallelic clones. We do try to identify clones. We do a lot of quality control, both molecular, genetic, and cell biological, to identify the best clones. This takes many months. And our cell lines are, all of our methods and cell lines are available on allencell.org. We want to study cardiomyocytes. Cardiomyocytes, of course, have very, very important proteins, such as those that localize and create the sarcomere, the contractile unit, that are not expressed when the cells are undifferentiated. So we had to develop methods to be able to actually do this type of approach in the context of having to screen visually clones in the undifferentiated state, and yet then wanting the protein that we want to tag be present only in the differentiated state. So we've done that. And both of these are papers that you can find. Here's a list of our cell lines. We have about 40 now that are available and more coming. What's really beautiful is that these cell lines have actually now exploded as far as I'm concerned. There's 600 of those vials that have gone out, and some of them go to cores, which is, has a much broader impact than just the single lab user. And so this is actually really exciting to us. We have, if you look closely, hopefully represented, at least in one instance, one protein that represents all of the main structures in the cell. For our legacy lines, we have a set of cardiomyocyte lines, and we are taking a deeper dive into the nucleus. And so a lot of our newer lines are really focused on trying to tag landmarks in the nucleus to be able to build an integrated nucleus as well, and together with the 4D nucleome. We have a couple of tool lines to start permitting perturbation of our system coming out soon, too. So what do these cells look like when we gene edit them? This is a colony of our cells grown on mesogel. The colonies are very tightly packed. They're epithelial-like in nature. They have an apical basal axis. They're very dynamic. And when you gene edit them and look at, say, tubulin, you can see the behavior of the individual cells in the colony much more clearly. These cells like to grow, divide, die, and then differentiate if you tell them to. It shows how beautiful our gene-edited cells look and how uniform the signal is between cells, for example. And here you can see a 3D Z-stack from the bottom to the top of the cell. You can see the microtubules. You can see the mid-bodies. And you can watch these in time, which we do in 3D. And here is a picture of 18 of our cell lines. Only the top row is going to move up and down in Z. And that's because our PowerPoint will crash if I do anything else. And you get an idea of what these cells are organized as. So this is sort of the picture of how many cells are in one field of view in this particular case. Here's some mitochondria. This is the top of the cell. Take my word for it. That's where the cell-cell junctions tend to live just like in epithelial cells. The organelles, the nucleus fills a lot of the cell. The organelles sit near the top. And if I do the one more animation I'm allowed to do without crashing the computer, you can see here proteins that tag the sarcomere and show beautiful beating cardiomyocytes. And what's really cool is actually, depending on which part of the sarcomere you tag, you do get different banding patterns, just as you should, which we think is just really beautiful. So these cells, of course, can be differentiated into more than just cardiomyocytes. So these are images from Bino Friedman at the University of Washington, who is using them to create mini kidneys, kidney organoids. And here you can see several of our cell lines used and imaged in that way. Here's tubulin again. And I could keep showing you examples, although we haven't seen all the data from all these examples, but we know that there are many, many people working now in many differentiated systems using our cells. We're incredibly excited by that. So we look forward to seeing where that all goes. OK, so I mentioned this holistic approach and wanting to think of the cell as a whole and have all of the parts in the cell. So how do we do that if we just tagging one structure at a time? How do we build integrated cells? So I'm going to show you 3 approaches we have very briefly. But this general approach to how we're then going to use those integrated cells will continue to be a theme that you will hear. So we can look at each of the cell lines individually, and we can actually do a lot with that. Just by themselves, we could analyze fields of view. We could understand how the structures behave. We can take time lapses. We create this assay for the undifferentiated cell. Starts with the membrane and the DNA as referencing systems so that you can pull out where the cell is and where the nucleus is, which we need in order to be able to do any sort of image processing to identify the cell and then to put structures in it. We have our scalable microscopy pipeline, which I know people sometimes laugh why I have this here, but it's actually quite a lot of work to bring it from R&D to production so that we can create really reproducible imaging. And then we try to create these models. Once we have an integrated model, which I will go into in a moment, we, even if we don't have it fully integrated, even if we just do it, for example, in parallel, we can really start to do an analysis of the variation of the cells that we're collecting. And we can look for contributions to that variation, sources of variation, such as the cell cycle or cell neighbors, and take that cell through states such as stress state or differentiation. And we're doing this for the undifferentiated cell, the differentiated cardiomyocyte, and as I said, this deeper dive into the nucleus. And again, we are doing this integration beyond just the spatial by including gene expression, and then as we're moving into the nucleus together with 4D nucleome, trying to identify good ways to merge structure and these biochemical assays to get at where the 3D genome sits inside the cells. So for the rest of the talk, I'm going to focus on this undifferentiated HIPS cell and its state, which is this sort of epithelial-like cell and has all of this potential to differentiate into many, many different cell types. So here's another view of this to just get you oriented. Usually, if I show you Z-stacks, they're going to move from the bottom to the top. The cells are very tightly packed. The DNA in those cells, the nucleus takes up 30 to 50% of the volume. And here you can see as you're moving up to the top of the cell, in this case mitochondria, the organelles fill up the cytoplasm and often looks like they're sitting more near the top because that's where space in the cell. So this is the integration assay. This is the data that if you go online you would get and that we've been focusing on. We have a dye to mark the cell membrane, a dye to mark the DNA. We have byte field, which will become incredibly important, and then we have one structure at a time that we image. This is a little bit of a snippet from our pipeline, just to show you that we have a workflow. We have a lot of workflows at the Institute. And we can go from edited cells to semi-automated and getting more and more automated sample prep and imaging, and then moving things into more and more automated segmentation and analysis. We have several wonderful workhorse modalities. We use the spinning disk heavily. We also have several ARIScan fast microscopes we love to use, and lattice light sheet is coming. We needed to wait till that was also a bit more turnkey and scalable, and we're looking forward to seeing that happen now in the near future. So again, this is a focus on an imaging pipeline that is live cell, 3D, and high resolution, which makes it a little bit different from other types of screening platforms. OK, so how do we create these integrated cells? Well, we have 3 different approaches, and they're all complementary to each other. We have an experimental integration, or a superimposition. So you can image the cells, for example, in all the stages of mitosis for all of the structures, and actually physically overlay them. And you can see that as the integrated mitotic cell story that we have online. You can use deep learning to train the machine to be able to make predictions based on all of the data that comes in, given a cell and nuclear shape, where structures might be, and then integrate them in that way, which gives you a beautiful statistical representation of and probabilities of what and how a cell might be organized. And we have the label-free approach, which the famous movie that I'm going to start playing here, which allows us to convert byte field images in 3D into predictions of fluorescence images of where the structures are, and that we are now extending to also predict the segmentations of where those structures are. And this particular tool, which is beautiful and blew my mind away, took my breath away, blew my mind when I saw it the first time. We also adapt it depending on our need case. So one place where, for example, we're really excited about it is in building the integrated nucleus. And of course, extending possibilities in time lapse. OK, so the rest of the talk is again on this undifferentiated cell and starting to take it through sources of variation and that state space. These are the 2 vignettes I'm going to focus on for the rest of the talk. This one I called, maybe boldly, towards a spatial, so an image-based single-cell assay. So if you want to be able to do your single-cell analysis in these cells, which are very 3D at high resolution, we have to pull those cells out accurately, and we want to pull all the cells out so that we're not biasing our sample. Then I'm going to show you an example of what we can do to start decoding the variance that's in our data and focusing on the nucleus. So just really briefly to introduce one of our tools, which Jianchu Chen is going to tell you more about, just 2 talks behind me, so I'm going to do that part a little bit briefly. We had this challenge that we wanted to segment out the cell, the DNA, and then each structure. And as we were trying to build workflows to do this relatively well, not perfectly, but as well as we could achieve, we found that that was very challenging when you have 18 structures like this, and they're all in 3D, and you can't just use one workflow or one method. One doesn't fit all. And so as we were doing this structure by structure, we started to identify common threads, common denominators, in which types of structures work with which workflows. And we thought, well, if we have that information, let's package it up in a way that others can use it too, if it happens to work for them. So that's, we developed a Python-based tool that lives in Jupyter Notebooks, which you can get at this link right here, which we call the Allen Cell Structure Segmenter. And it has 2 parts. The classic image segmentation workflow, Jianchu will tell you more about that, and a iterative deep learning workflow that takes the output of this classic workflow to create training sets to create deep learning models for segmentation. So just really briefly, this is a little bit more information on the classic workflow. We have several different very classic algorithms. It's not the algorithms themselves. It's the chaining together of the algorithms that matters. Our output right now are 3D binary image stacks, which really falls more into semantic segmentation. That's something that may not be useful for everyone right out of the gate. So getting the detection of where all the objects are in the entire field of view. But we're going to be, it's quite easy to do this, say, in ImageJ, to convert that to instant segmentation for some objects. And we're going to be trying to incorporate that into Segmentor so that you get obvious objects or all structures in, for example, a masked cell, and so on. Once you create these workflows for each of the structures, we have this lookup table, which we created to help users see which workflow they might want to use. And Janshu will tell you more about that. I want to briefly introduce you to this iterative deep learning component, because it's been incredibly important for us in what we've done recently. And its basis really is that if you get a relatively good output from your classic segmentation or from any manual annotation that you do. And I can tell you that I have manually annotated complex shapes in 3 dimensions for a total of maybe 5 cells and never again. So this is something where I would much rather tweak those parameters in the segmentation to get something to work well. And it doesn't have to work perfectly, and that's really the key here. We're not creating the perfect ground truth here. We're going to take it through a sorting and merging step to create the ground truth to build the model. And this allows us to improve the accuracy and, very importantly, the robustness, because what we do here at the Institute has to scale. So if we take lamin B1 here, which in interface outlines the nuclear envelope beautifully, you can see that in mitosis it doesn't look the same. And the segmentation that is going to work really well for interface nuclei is not going to work so well for a mitotic distribution of lamin. But this particular classic segmentation workflow that we used also missed an interface nucleus here, which is what can happen. So the first step you can do is just, by chance, take serendipity, take advantage of chance, and keep those images where your detection and segmentation of, say, these nuclei worked. And then we actually, in this case, only kept 8 and trained a model based on 8 images, 8 3D images. And from that, already got a result that greatly improved what we did, which I'll show you in a second. If we then want to be able to incorporate, say, a different distribution type, or if you're using transfected cells, different intensity levels or different morphologies, or whichever way, whatever it is that your sample has, you can now mask out, for example, 2 or more regions of an image and use different segmentation algorithms within each of those regions and merge them to create a merged ground truth, which means that the model will now learn the full variation that you have in your images and the best segmentations for each of them. So when we do that here, first with the sorting, we improve our interface detection greatly. We've picked up that nucleus now. And then once we do merging, we can get the mitotic detection that we want. Janshu will tell you more about how to actually use this. So now our goal is we have something for the structures. How do we get all the cells and all the nuclei in an image in 3D? In this case, where if you watch really carefully, these cells have wild shapes. I mean, they're tilted and leaning on each other. And you don't want to attribute parts of an organelle from one cell to the other. You want to try to get these boundaries clean. And you want to do it accurately. So as many of you know, if you've imaged anything solid, if you image it in 3D and you image it solid, you have a very hard time seeing the boundary in Z, because of course you have poor resolution in Z and blurring in Z, and you just can't tell where the boundary is. So this here is XY, and here's Z. And here is the DNA dye, and here is our lamin signal, just to show you that it's actually quite hard to know right where the boundary should be for this DNA. But more importantly, this is our assay. Right? This is the quality of the data that we want to pull the nuclei out of. This is the quality of one of our cell lines. Wouldn't it be nice if we could take that information and take it into account as we create the segmentations for what our assay is going to be? And we call that a training assay. I think you've heard examples of that actually at this talk. We've sort of formalized that wording for ourselves. This is true for the membrane too. We can see, and this is actually from the side again, so this is the bottom of the cells, and this is the top of the cells. You can see these nice shapes and leaning and overlaps that the cells have, and you can see them quite well in this cell line that has tagged the membrane. But you don't see that so well with the membrane dye uniformly in all of our cells. So again, it would be really great if we could transfer that information over. So we can do that. We can train a segmentation model for the membrane dye with the knowledge of our CAAX signal. So we create a classic segmentation based on the cleaner CAAX signal for these cells. And this also requires the sorting step, because any of you who've tried to do this know how hard it is to actually get accurate boundaries that are where you want them to be. And you get a CAAX segmentation model. You take that model and you rerun it on your CAAX image, and you get a CAAX-based ground truth, which you then actually combine with the membrane dye to then create your membrane segmentation model. We do the same thing for the DNA, and we take into account mitotics by using the merging. And this is the result that we now get. So the model output is here. It needs to be refined, right? So you have to go from semantic to instant segmentation. So we use the membrane to cut the nuclei. We use the nuclei as seeds to detect all of the cells using a watershed. And in 3D, you can see that the segmentations are absolutely beautiful. And when we did an accuracy inspection over 180 images over our whole pipeline, we can get incredibly high numbers of individual cell and nucleus accuracy, but much more excitingly, entire fields of view, which really allows us to start doing an analysis on all of our cells. This is incredibly exciting. This was a technological hurdle for us to do the type of analysis we want to do. And we are now in the process, literally running it, or have in the middle of running it, to test it on the 15,000 fields of view that we have in our pipeline. And we will see how much more of this we need to do to make it scale. So some of these steps for scaling become really, really important when you do this at scale. And we're ready to assess how well it's going and start using the data. So that's great. But we actually think this is a generalizable approach to get 3D accurate segmentations of cells for others in other systems. And so this, all you need for that is the segmenter, which is out there, and this concept of a training assay, which I just told you, and you probably already know. And so these are questions, does it scale? We're working on that. Can we do this on other magnifications? Can we do this via label-free? Can we finally do time-lapse because the segmentations are robust enough that we don't have to somehow figure out how to curate time-lapse? Is it robust to perturbations, and can it work for other cells? These last two I'm not going to address, but I hope that you will. So it would be really great if we could extend our segmentation detection approaches just directly from bright field. So can we use our label-free UNet model that we have to try to predict not fluorescence images, but specific boundaries of the cells in the DNA? And the answer is yes, with varying accuracy. So this is for the DNA, and it's beautiful. And it's almost as good as the dye segmentation. So we're very excited to start doing that all via bright field in 3D. And this is the result for the cell boundary, which isn't terrible. You can't go from this to this. It's pretty amazing. If you look at the original, you will see that the model has a hard time at some of those very particular cell-cell boundaries that have funny shapes. So we're actually improving the model to try to solve this. And then we hope that we'll be able to actually switch to using bright field as our signal and get away from these pesky, terrible dyes that cause all sorts of problems. So does it generalize to label-free? Yes. OK. I'm going to answer some of the other questions as we go into the actual scientific question that then motivates us to test those questions, brings us into time-lapse, and brings us into some other things. So we want to build a state space. And to do that, we have to understand which cells are more similar to each other and which are not. And we have to start decoding the sources of variation in our data. And what I mean with that is, for example, if you watch the cell, and I'm focusing on the nucleus for this part of the talk, OK, but this is going to extend to all of our structures. If you look at this 20x movie that's 13 hours long of the lamin B1, cells doing their thing in 3D, you can see that one of the things, of course, matters dramatically if you're going to compare two cells to each other is what stage of the cell cycle they're in. If they're in different parts of the cell cycle and they have a nucleus that's twofold larger than the other, you don't want to compare those two cells and then look at all the other differences. They're not. That is one source of variation you want to account for. Other sources of variation you want to account for are cell neighbors. I stopped playing this movie so that you don't just stare at that movie and get totally distracted by everything happening in it. But hopefully you saw, while the movie was playing, that these nuclei were pushing on each other all the time and changing their shapes. And so the specific set of neighbors and their impact on a particular nucleus is going to affect what that nucleus is doing and how you should consider what that nucleus will be doing. So that's a source of variation you have to take into account. The position in the colony will matter, and you'll see more about that. And the behavior of the cell or in the nucleus in time. And what I mean with that is how ergodic is the system? Is it, so ignoring now the neighbors, just that nucleus on its own, does it explore all of the parameter space that it's possible to explore, or is it sort of on a path? Is it a reproducible path from nucleus to nucleus? Is there more variation within the trajectory of the nucleus than between nuclei? These pieces of information are really important to being able to interpret the sources of variation if you compare two nuclei next to two nuclei that are similar in every other way. So let's say that we've now done that. We've taken two nuclei, their neighbors are similar, they have the same sort of behavior in time, they're in the same position as the cell, and now you compare the nuclei and the cell and you start seeing differences. Now you're probing that next level of variation, which could be other naturally occurring states. One cell might be more starved or more stressed. And it's really, really hard to start picking up those differences and identifying what is normal and what is different if you don't take it all into account. So this story is about trying to do this for the nucleus, at least as a prototype, for what we want to do to analyze what we call intracellular morphogenesis. How do these structures form? What is the variation in those structures? And how do they change? And how do they then interplay with others? So we want to identify these sources of variation, starting with the nucleus and the cell. We want to expand this to other structures. So here you can see similar types of movies. So you can see the behavior of other structures sort of in that framework. And we want to incorporate that integration, that holistic approach, and the dependencies between structures into that analysis. So what I'm going to show you is a prototype based on the nucleus that we're now going to do for many other structures. Next is the cell, and then the nucleolus. So for that, we need an accurate and robust segmentation of all the nuclei and all the cells. So now you know why I was talking about that. But we don't need it in our pipeline data. We need it there too, but we now need it in these types of data. So let's say we said, well, this should be doable for lamin B1. Let's start with the most tractable structure. We have a great segmentation of lamin. Others have great segmentations of lamin. This should be robust enough to be able to do. So let's take this 20x movie and do that. Oops. So we set out to do that, and we started looking at our lamin, and we saw that we just can't tell at 20x of a 0.8 NA objective where the top of the nucleus is. And we want to analyze the shape of the nucleus, so that's not so great. So we decided, let's just do a compromise for right now. We're going to come back to this point, but let's do a compromise and see if this type of analysis works and what we can find. And let's do this at 40x. So that immediately says, well, can we do our segmentation at 40x? And will it then be good enough to do this in a time-lapse manner? So there's a bunch of information about this movie. I will probably go through it in a moment. Right now, I'm just showing you the first image of that movie. It's a 40x movie at 1.2 NA with the water objective for 6 hours. And this is a segmentation that we've retrained on the 40x lamin. It looks really nice. So it does, here's a Z view. It does generalize very nicely. So now we take that movie and we play it in time. Before I hit play, I want to point out that the little dot in the center is the 3D centroid. This is tracked in 3D, not in 2D. The boundaries are also actually 3D. You're just seeing max projections. Because they're max projections, some of the segmentations may look more or less good because of the way that we're visualizing them. And we are not yet worrying about mitosis. We're going to solve the mitosis problem using our H2B line, where we can track the DNA all the way through. The tracking is detection association. And here is the movie playing. And if you watch some of these nuclei here, you can see that, to me, this was awesome. We did not have to go and curate any of the tracks, except for the very, very, right when they go towards mitosis, or right when they come out of mitosis, they're incredibly accurate and robust. So we could now filter this data and say, OK, well, we'll just start an analysis of interface just to see how this looks. And so we're going to keep only interface tracks that are longer than 45 time points, which is 75% of the tracks. So we have a final data set of 65 tracks, which ended up being 4,600 nuclei that we then analyzed. Oops, I'm pointing the laser pointer is not turning off. There we go. OK. So the approach we wanted to take is that now we have to find a way to quantify the nucleus in some meaningful way. And we decided, well, the first thing we have to do is be able to fit the shape of this sort of semi-spherical structure. So we decided to apply spherical harmonics, which are, of course, useful for describing continuous functions on the surface of a sphere. They are indexed by order L and integer m. And they can be used for fitting complex shapes. And depending on how many how far you go in the order number, you can get different sort of aspects of that shape and different extent of fitting for that shape. So each combination of L and M has a coefficient value that describes its contribution to that fit. And we thought, well, we'll take those coefficients and we'll start analyzing them as feature parameters. So here's just to show you, first of all, can we fit our segmentations of our nuclei to the spherical harmonics? And here you can see a goodness of fit. And after order of about 4, that doesn't improve very much more. And you can see that it actually looks quite nice. And then we plotted here the first 5 different coefficients. and the magnitude that they have and the cumulative magnitude. And what we started doing is we started using all of the coefficients to do a PCA analysis. We started doing correlations between some of the features that we knew described shapes in our nuclei and these coefficients. And it turned out there's actually a one-to-one between some of the key coefficients and what aspect of shape, what mode of shape they describe. And the PCA analysis ended up matching exactly just the magnitude of those coefficients. So if you want to know more about that, come find me. It's actually really cool. So the first one is the volume. That means a smaller versus a larger nucleus. This is the x-axis, the y-axis, and here's again the axial view. The second one is the flatness in z. The third one is how squeezed it is in xy. These are actually very similar to each other. Are they more or less flat, along the apical basal axis, and are they more squeezed by neighbors? So now we can take this data and plot it. This is time on the bottom. We just took every one of our traces, 65 traces, and just plotted them here for, say, volume, or this should say z flatness, not squishiness. I changed and this should say squeezing. I will change that next time. And we just highlighted 3 random cells that had a small, medium, and large size nucleus at the start of the movie. So these are not aligned to cell cycle. These are just the raw traces from the movie. But they allow us to just look. So for example, over 6 hours, a nucleus that's small at the size that when a nucleus is born doesn't reach all the way to the size it is when it divides, because it's only 6 hours and the cell cycle is 24. So we need longer trajectories to get the variation due to the cell cycle. But what we could see is that this flatness in z varies much less within its trajectory than, say, the squeezing in xy. And this little hump here actually was a particular cell that had a mitotic and an apoptotic cell near it, both squeezing it really intensely in one moment in time, and then it relaxed again. So this actually takes a long time to explain, so I'm going to do it not as slowly as I would like, but this is actually this is not x, y, z space. This is the 3 different modes. And you can actually do visualizations to start watching a particular nucleus in time as it moves through that shape space. And you can line up the trajectories in that shape space and start looking at specific, so this is all the cells, and then we're going to highlight one particular trajectory. And this allows us to start thinking about using things like the percolation within this space as a measure of the ergodicity of how much of the variation of the whole population these nuclei experience. So what we see is we have 3 different sort of shape modes, which actually end up mapping to 3 different sources of variation: the cell cycle for the nuclear volume, squeezing in xy, an order of minutes, and then something which I'm going to tell you about a little bit more now, which is z flatness, which actually is on the time scale of days because it's due to cell packing and how the colony behaves. So if you look at fields of view of flatter nuclei or rounder nuclei, it looks like you have shorter or taller cells. And it also looks like you might have denser packing, which we tend to see on the outsides of the colonies. So one of these very, very important modes of variation, the second mode of variation, which was the z variation, actually comes from this colony behavior over long periods of time. We wanted to check that by actually taking our production data set and looking at it. So taller cells have less flat DNA, and they have greater density. So it's exactly the way that we would suspect, and that as an experimentalist, I and my experimental part of my team would have told you just from looking. But it's very nice to be able to quantify it. So next up is that we want to actually take advantage of these sorts of data to create mechanical models of nuclear shape and colony dynamics, but we also have challenges which are beautiful to try to solve. This is actually what the nuclei do in time. The lamin doesn't show you that. They're tumbling in all 3 directions. And we have to think about how that affects our interpretations of the shape and the modes of shape. So that's next up. And this is a nucleolus that's just letting you see the inside of the nucleus. OK, so we want to be able to expand this to the cell and to other structures. Here we go. Can we do that? Well, the first question is, can we do spherical harmonics on a cell? So here's an outline of a cell, and you can see the fit, and it works quite nicely. So now we can actually take cells, and at least for these 3 structures where spherical harmonics make sense, we can do that on the cell shape, the nucleus, which you've already seen, and the nucleolus. So that's where we're going to start, since we can use the exact same approach and explore this a bit more. And then we'll change our approach if we need to as we go to things like mitochondria. Oh, and here's the fit, sorry. It works very, very nicely. So we go back to our problem, which is that we really want to do these in these 20x movies. And that's because we can image over 2 days for those in 3D. We can, even with 40x, we can maybe get, we can get to 6 hours, maybe a bit more, and it gets cumbersome, and things start bleaching. And of course, at 100x, we don't get very far. So the question is, can we actually take these images and transform them into these images in a quantitative way so we can use the data? So basically, can we take something that's at a 100x 1.25 NA magnification and resolution and field of view like this, and expand that field of view and maintain all of that information? And so we, as others, thought, well, maybe we can try some deep learning on this and see how well it works for our quantitative needs. So we teamed up with Danny Chen and his student Hongxiao Wang, who worked with us this summer. And we said, OK, let's think through this. Let's start with lamin, because that's the question we're actually asking. And let's take images at 100x and at 20x that we've imaged in fixed cells, exact same cells, align them exactly in space, and then train a conditional GAN, a CGAN, in order to see if we can then predict out from this information something that looks like that. And so we tried this with a couple of different backbones. And our result comes out like this. So that looks good, right? Yay. So we can look at it a little bit closer. The yay will come. I will give you the real yay in a moment. But the point I'm making is it looks nice. We look at it more closely, and it looks beautiful. So we say, great, let's try this on something that is a little bit harder, that has texture. So we're really interested in the nucleus and in mapping the heterochromatin and other structures in the nucleus. So let's try histone 2b. Here's our target, and here's our prediction. And it looks quite good. And you start looking, you start seeing slight differences. So we're going to validate and work on this some more. But really importantly, this looked great, so we said, let's use it. Not so fast. So we said, OK, let's take our predicted and our original. So this is the original 100x, and this is the predicted. And let's actually pull out, segment out our nuclei and look at them and see if they're the same. And the answer is they're not. They're about 8% off in a very systematic way. And when we started using synthetic data to try to figure out where that was coming from, we realized this is actually because of a Z alignment problem, right? If you take two samples and you're imaging the sample in two different settings, you are minimally going to have an error that's due to the size of the Z step, because you can't center both in that same exact pixel, voxel location. But we can try to fix it some other way. So we actually added in a module that allows us to learn that difference and correct our images for it. And now we could take this quantitative plot and turn it into this one, where we have apparently something happened. This is 0.98 now. From 8% error to 2% error. And we got the similar results of all of the main spherical harmonic coefficients we care about, so not just volume. So that really does excite us. So we actually can start doing that now. And we said, well, let's use that same model and try out some other important and relevant transfers for us at the Institute. And one of them is to switch between modalities, since it's really hard to always create all of your quantifications for all of the different modalities over and over again. And of course, because we want to enhance our resolution if we can. So we trained a model to convert 100x spinning disk images into area scan fast images, which works incredibly nicely. And then we said, well, can we daisy chain those models? And so we're still in the process of validating this. This is a first look, but very excited about it. So if we start with a 20x image and predict up the 100x image, and then we predict it to an area scan fast image, we get something that looks like it's working quite well. Of course, the devil is in the details, and I just showed you that we care deeply about those details. So we are not going to say this is working yet until we actually validate it carefully. We're collecting the data right now to have the proper ground truths to compare these. And we're really excited also to see whether a direct prediction versus a daisy chain prediction has different accuracy, and what the limits are of what the model can do going forward and moving backwards. So what we can do now is we can take this image of a slice, a time point in a movie of our 20x data and convert it to segmentations of nuclei based on predictions to 100x, which are in 3D and which already show us some of the trends we saw before. So that you see taller nuclei in regions that are more densely packed. And we can do that now fully in time. So this is our 13-hour movie, fully segmented at 100x resolution accuracy. And this highlights for us that as we go from image to information at the Institute, we really need it to be quantitative. We actually want to use it for what we're doing, so we have to be very careful how we validate it. Robust, because we want to scale it, and accessible, meaning that hopefully we'll get that to you guys within our segmenter as soon as it's ready. So I just have 2 slides to finish up. I told you about the undifferentiated HIPS cell. I don't want you to forget that we're really working also on the process of differentiation to the cardiomyocyte. So here you can see some of the images we have through time. A much longer time lapse. This is not a time lapse. This is different images over 12 days. And also that we're taking a deeper look in the nucleus together with the 4D nucleome. And so here's a smattering of structures within the nucleus itself, so the nuclear periphery, the nucleolus, the speckles. These are actually transcription factories. This is pCNA, which marks sites of DNA replication, and these are telomeres. So now we're going to try to take the same approach, both for the cardiomyocyte and for the nucleus, which now requires a different referencing system, which we are now developing based on these structures here and incorporating label-free. So I want to thank the whole Allen Institute for Cell Science. We don't tend to pick out very specific individual people because we all work on all of this together as a big team. It's actually one of the things that makes it so wonderful to work here. So every single one of these people contributed to this talk. And so did our founder, Paul Allen, who I also want to thank for his inspiration and vision. Thank you very much.
Thank you, Susan. I'm quite sure there are a lot of questions. Please wave to Callista or to Melissa to get the mic when asking your questions. One is back there.
Susanne, thank you very much. I have two probably long answer questions, but you can do a short answer first. First thing is as I mentioned yesterday, right? Pathologists look at nuclear shape.
Yes.
And what you now showed so far is there's geometry, there's cell cycle. Those are probably relatively neighboring cells, and cell cycle. Those are relatively simple effects. Have you guys some indication that there might be now some molecular upregulation or whatever, right, that goes into this nuclear shape? And then the second question is, as you go into differentiated cells, do the spherical harmonics still work in classifying cell shape? Because you have relatively globular cells, and that's clear. Spherical harmonics work beautifully there, right? We have known that for 200 years, right? But as shapes now get more complicated in the differentiation process, where do these things break?
Okay, second one first. So it turns out our cells look globular the way we show them to you because they're all packed to each other and because of the dye. And so the cells actually, if you isolate an individual cell and have just one cell tagged in the sea of other cells, because they can't, they're not happy by themselves, they actually have beautiful protrusions. They look like little octopuses. And we have actually tried the spherical harmonic fit on that example cell, which was not even, this laser wants to kill you guys, was not even at high resolution, and the spherical harmonic fit looked quite nice. I can show it to you. That does not mean it will extend to all cell shapes, and we did not actually presume to. We actually were using this just as our in-house way to quantify, to start doing this type of analysis. The second question, which is, are there molecular indicators of how and where these modes of variation come from? I think there will be, but right now we're still at the level of decoupling the really big modes of variation that are so obvious, and they're very mechanical and physical, that we're not there yet. But that's where we're trying to go. And the nucleus, of course, is a beautiful target, especially combined with the nucleolus, because of that pathologist application. It may be that it's harder to figure it out there or not, depending on how it responds to things when we induce certain types of stress, where we know that we're affecting certain signaling pathways and such. So that's coming, but we don't have that information.
Beautiful talk. Thanks. So you mentioned 13 hours of movies. So just a technical question on how much do you think ROS you generate? How often are you taking the images? Is it 20% oxygen? Is it 3% oxygen? And how do you think if you need a movie over 7 days or longer, how many cells you're killing or affecting?
So we actually,
process of observation.
So our cells, as you heard yesterday, our cells are incredibly sensitive to phototoxicity,
Exactly.
especially at high magnification, and especially once we add dyes. And so we actually had a photoprotective cocktail we use in those circumstances to help, and then we have to start worrying very much about ROS production and how much we're protecting it and whether we're inducing hypoxia, which we are, and so on and so forth. The images I showed you here, when we image at 20x, the cells are incredibly robust. I'm sure they are producing ROS, but we see none of those phenotypes that we're incredibly used to dealing with at higher mag. And so the almost 2-day, the limit there is not the imaging. That was at 5-minute intervals for 2 days at 20x. And these cells, again, are sensitive. But we can just go down so much with the light. And of course, our goal is to be able to then do this with label-free and even, for certain structures, even skip the fluorescence entirely. And that's what we're working towards. But the 20x system, if we can scale it up to 100x, well, even if it's not for all structures, is really powerful for time-lapse for us. So.
Thanks.
Are there any? Okay.
Thank you for the great talk. So I'm kind of from a different perspective because I'm a researcher in machine learning. So in machine learning, there are deep learning people, non-deep learning people. I think deep learning people are usually criticized by non-deep learning people that you treat deep learning as a black box. Do you know how it works? And if you don't know how it works, and when it applied to domain, people will have serious concerns. So my question here is, you used a lot of deep learning in your context. Do you think interpretability is needed, or is needed in your context? If it cannot be interpreted, is it going to be a concern in the future?
It is completely context specific. So in the context of image processing, where the output that you get is something you can quantitatively validate, if you think about it carefully, the way I tried to show in this, each particular application would have its own type of validation. I think it does not matter because the validation will speak to it. And as long as you show that that validation scales for all of your data and that you're comfortable, you will be able to convince me, who is the most skeptic biologist you're going to find on this particular part, that it's OK. But if you start doing it, and this is something we're facing, we're doing it very carefully. If you start wanting to make biological interpretations on things that are hard to see, that are based on deep learning, then those questions about black box will come up. And I think that the answer there is that you have to think about the right way to do the validation for those models back to what you know about the biology and what the models predict, to start to either start deciphering the black box, which I'll come back to in a second, or to just give people that certainty that it's OK. And for the deciphering, what I mean is that Julie Theriault actually took our integrated statistical model framework that Greg Johnson developed here at the Institute and applied it to her neutrophils, which she knows a lot about how they behave, and actually started investigating the latent space and the latent space versus the PCA space that she's lived in, and found really, really interesting behaviors within that black box that actually might be extractable and usable. And so she and others, and you all, hopefully will start venturing into that as its own sort of part of the deep learning field, that maybe there is actually information to be pulled out in a useful way. And that type of thing would, of course, help biologists feel more comfortable. But I think it's very specific and context specific. So.
Thanks for that talk. I really like how you're leveraging your low ambiguity images to try to get more information out of your lower resolution objects. When you're applying that and you're using those models, how do you protect yourself from making the wrong inferences from those low resolution objects? I mean, that is to say, are there process controls that make sure you're feeding the right data into the model?
So right now, this project is quite new. And all of our validation, and so I care deeply about these things. So, all of our validation right now is based on these fixed images where we can have exact one-to-one matches between the modalities. And so we hopefully have, the only ambiguity we have is the limits of the resolution of the best version of the modality that we can have. Right? We can't get better. If our target is 100x spinning disk, we're not going to get better than that. But we can certainly match to that, and we can certainly think about ways to then ensure and quantitatively test in the context of the measurements we're trying to make whether that's working. So for us, we feel like if we can have a particular data set in one version, and then that same data set in the other version, and the biology that we're asking comes out the same, or with some error, then we start to feel confident in it. And that's going to be actually a really reoccurring theme in validation of biology, is that if you can give it an error bar in the context of the application you need, you're probably OK. But that does not mean you should suddenly go out there and generalize it to everything else.
I have a question. I think this is a beautiful talk, but I want to actually ask a question about all the computed results. Okay, even after validation, okay, if they kind of match very well with the training data sets, certainly that could be viewed as a success. I know that you mentioned this is a new project, need more validation to be done. I think that sounds very good. Okay, sounds like a pretty promising. But my question is not about that. It's more than just validation. I think actually the whole purpose of why we are kind of doing all this kind of biological large-scale project actually is really try to use the kind of integrated way, the synergized way to let us be able to see the biological truth. Okay. My question actually is that, okay, from all this kind of effort, okay, what will be your vision? How we are going to really get this biological truth, right? So, when you say, okay, the distribution of the shape, what are the actual true pattern out there? Okay, it's more than just the computed result, right? So yesterday, Gondes actually, mentioned, based on the lotto screening, generate the modeling, and then eventually come up with some kind of modeling about this causal relationship, I think that's a very nice way to at least a very nice effort to go that direction, okay,
Correct.
to find out the biologically true pattern, okay. I think many interesting things, but I want to know actually from the cell science perspective, this institute, how you guys are going to really get this biological truth out of this tremendous amount of beautiful data.
Yeah, so that's a good question. We are figuring that out as we go. We believe deeply that we are not just collecting data for the data's sake, right? We have very specific questions we ask. We want to, for example, see what patterns distinguish cells. So this is a longer answer actually, but do I have time to go into this?
Yes.
Yes, great, it's important. We want to, for example, be able to say, we're going to start slow, we're going to build up. So we're using this nucleus as a prototype to do this for all of the structures. So in the case of the nucleus, we're going to say, what is the pattern that we see for the nucleus shape, and where can it come from? So there's two places to it. One is on the image side and the analysis that you saw here that says, okay, what are the sources of variation? Are there pathways that might impact it? That will become interesting. Do we see, once we decode that variation, have the underlying shape that we care about in the particular context? Is that its own state? Are all our cells clustered there or not? Is it affected when we move them into some other state, like a stress state, which we're actually developing assays for? And which of the structures do so? Which of the structures respond to the particular variations in nuclear shape, for example, or the particular variations in cell shape? And can we pull out dependencies between those structures that come from that type of information? The other approach is actually from biophysical modeling, for example. In the context of the nucleus, I'm just going to use that as an example. If we have these metrics for nuclear shape and how it changes in time, we can start actually formulating models of what the mechanical properties and the behavior of the nucleus is, and match that to what the shapes are, and create hypotheses, for example, in the context of just the cell cycle and the nucleus growing. How does that even happen? What are the mechanical needs you need for that? And does that fit with the type of data that we now pull out? So the challenge to your question is that in order to build the big picture answer, you need all those little pieces all coming together. But we're now setting up to say we're going to prototype that concept with this approach, which we're starting on the nucleus. And we're integrating it with the other structures because we can get the nucleus in the cell for all of our structures. And then we're going to see how that comes out. So we don't know that it will 100% work, but we firmly believe that there will be a lot in there that we and others hopefully have not looked at in that way because of that integration between the structures. And they're looking at, does a slight movement, OK, here's a null question. The nucleus fills the cell, you have the cytoplasm. Is everything else just fill the cytoplasm evenly, or is there some order to it or some structure to it? Well, first we have to be able to pull out that cytoplasm. Then we have to start looking at those structures and see how that happens. And then we can do that in the context of where the variation is in the shape. So we do think it's going to be there, and that's how we find patterns. But it's not going to be, it's like Gaudens said. So Gaudens is a great inspiration on this. A, because of these very, very careful ways of trying to get a causality. And B, that wonderful thing that you guys all believe and we believe too, is if you have a new way of looking, you will see things you can't see otherwise. So we do believe that that's going to happen.
Yeah, thank you for this very interesting discussion. Thank you, Susan. So now.